Good day, everyone. My presentation for today is entitled Visualizing Life, Turning Biological Data into Discovery with Artificial Intelligence. Today, I will focus more on a very practical question. How can artificial intelligence help us move from raw biological data to a clearer patterns, better visualizations, and more meaningful interpretations? My goal for today, for this discussion, is not to promote one particular tool, but rather to show a responsible workflow that teachers, students, researchers can use. So, let us begin.
So we all know AI can simulate human reasoning and problem solving. But in our context today, I want you to think of it as an eager, incredibly fast, but kinda literal-minded lab assistant. It's not magic, it's just a tool, a highly effective tool that we can use to process complex biological information and totally scale up our analytical capabilities. And this digital assistant really empowers you in three distinct ways: analyze, assist, and generate. AI isn't just one monolithic thing. It supports us biologists by finding hidden patterns, helping out with daily workflows, and actually creating entirely new outputs. Let's break down how those three work together. Analytical AI is your pattern finder. It's perfect for something like detecting trends in plant growth across different environments. Then, assistive AI steps in for the grunt work, helping clean up messy data sets or recommending the best statistical approaches. And finally, generative AI actually creates the deliverables. It generates the code, the visuals, and the plain language explanations to take you from raw data all the way to a finished visualization. Okay, this is an absolute game changer. Instead of staring at an intimidating raw statistical output, something like r equals -0.76, AI can instantly translate that math into an accessible biological explanation. It tells us directly in plain English that there is a strong negative relationship where dissolved oxygen decreases as water temperature increases. It completely shifts your focus away from calculating stats and gets you back to actual biological reasoning.
The sheer breadth of what AI can actually do right now is just staggering. It's really not just a niche tool for computer scientists anymore. It is touching literally every corner of biological science. Just think about it. In ecology, AI is out there forecasting outcomes and predicting species distributions across entire ecosystems. In image analysis, it's detecting and measuring things automatically, like counting cells so you don't have to sit there and squint at a monitor for hours on end. Whether you're analyzing genomic variants in bioinformatics or predicting plant growth, AI is acting like this massive accelerator. It translates raw biological data into actual, workable insights in a fraction of the time. Think about the old way for a second, the traditional workflow. To get anything done, you had to be a master of complex software, right? You had to memorize endless menus in SPSS or write flawless bug-free code in R or Python, all while perfectly executing these incredibly complex statistical procedures. Honestly, it was a massive barrier to entry. But the AI-assisted workflow, it completely flips the script. Now, the primary requirement is clear natural language interaction and your own biological intuition. You don't need to speak Python fluently anymore. You just need to speak biology clearly. How cool is that? When we compare these two workflows side by side, the traditional journey is an 8-step linear grind, from the research question to collecting, cleaning, selecting methods, analyzing, visualizing, and finally interpreting and reporting. It relies so heavily on technical software skills right in the middle of the process. But look at the new AI-assisted pipeline. It's actually expanded to 9 steps, but the real game changer is step 3, natural language interaction with AI. This is the new bridge. Instead of wrestling with code to inspect data or generate graphs, you literally just talk to the AI. But, and this is a big but, pay close attention to the final steps, which are human validation and biological interpretation. The machine does the heavy lifting, sure, but you hold the steering wheel. To really hammer this home, let's look at how your daily tasks actually change. Instead of manually inspecting variables or hunting down missing data yourself, you just ask the AI to describe the dataset and check its quality. Instead of spending hours researching which statistical test to run, you ask the AI to suggest possible methods. But here is absolutely the most important takeaway when comparing these tasks. When it comes to interpreting the results, the AI-assisted method still says researcher still interprets results. The AI acts as your coder, your fancy graphing calculator, and your proofreader, but it is not the scientist. You are. So you now have 6 specific superpowers at your fingertips the second you upload a raw dataset. They are understand, inspect, summarize, analyze, visualize, and explain. Think of this as your new analytical roadmap. It takes you seamlessly from raw data to meaningful interpretation, basically acting as an incredibly powerful assistant at every single station along the way. First up is summarizing. Before you can find any profound meaning in your data, you just need to know what it looks like, right? Well, AI can instantly compute and organize your basic descriptive results. We're talking frequencies, means, medians, standard deviations, ranges, and group summaries. It instantly calculates all these descriptive statistics, taking that manual, error-prone busywork entirely out of the equation. This means you can immediately start seeing the overall pattern of the data without pulling your hair out. And how do you actually get those summaries? Well, you just ask. If you're studying plant growth, for example, you literally just type this prompt, summarize plant height according to fertilizer treatment, and that's it. No writing complex array formulas, no digging through a million drop-down menus just to isolate variables. You just speak to it exactly like you would a research assistant, and it instantly generates the descriptive tables you need. It is incredibly freeing, let me tell you. Okay, so once you have your summary, you naturally move on to analysis. AI can suggest or even execute some really sophisticated statistical approaches. It can run comparisons, calculate correlations, handle complex regressions, classification, or clustering. But, and this is a very big but, AI should always suggest an analysis with a solid justification. It shouldn't just automatically select a test behind the scenes without telling you why. You need to ask it, hey, why are we using an ANOVA here instead of a t-test? And critically review its logic. The execution is automated, sure, but the judgment remains 100% human. Now, for many of us, visualization is the absolute major advantage of generative AI. Humans are visual creatures, right? We understand biology through patterns we can actually see. AI can instantly take that complex analysis we just talked about and generate bar charts, line graphs, scatter plots, histograms, box plots, and even super complex heat maps or interactive visualizations. Tweaking the colors, the axes, the labels, tasks that used to take a solid hour of tinkering in R, they now take a 5-second text prompt. It's a total game changer. Okay, I know it sounds a bit like magic at this point, but here is the crucial warning we have to talk about. AI absolutely cannot guarantee that its interpretation is biologically correct. An explanation generated by an AI might sound incredibly convincing. I mean, it'll be written in perfect English with a super authoritative tone using all the right jargon, but it can still be entirely scientifically incorrect. This is the danger zone, folks. To understand why it makes these mistakes, you really have to understand how it works under the hood. Generative AI operates on something called probabilistic generation. It doesn't actually know biology the way you do. It creates outputs based on statistical probability. It's essentially just predicting the most likely next word in a sentence based on its training data. Because of this, its outputs can contain fabricated or totally inaccurate information, which we commonly call hallucinations. It's doing math with language, not calculating absolute scientific truth. Therefore, your critical evaluation and human oversight aren't just recommended, they are strictly, absolutely essential. How will you balance this newfound analytical speed with your own critical scientific oversight? Generative AI has removed the friction of code and complex software. It has essentially given you a turbocharged engine for processing biological data. But remember, an engine doesn't know where the car is supposed to go. You are the scientist. You provide the intuition, the critical thinking, and the ultimate validation. Use the AI to completely eliminate the grind for sure, but never, ever let it outsource your scientific judgment. I want you to consider this quote for a second. Statistics quantify the pattern. Visualization lets us see the pattern. Far too often, we treat graphs like they're just pretty pictures, right? Something we slap on at the end of a report just to break up a wall of text. But think about it. While a p-value or a standard deviation gives you a vital mathematical summary, your visualization is actually your primary analytical tool. It's the literal lens through which you perceive the underlying biological reality. Good visualization really does the heavy lifting for researchers. Imagine you're tracking a new medication's effect on cell growth. A solid graph lets you seamlessly detect overarching trends, compare your experimental groups, and immediately spot those subtle variations or crazy outliers that might otherwise totally skew your statistics. It helps you examine relationships and discover patterns that are just hiding in the noise. When done right, visual design makes complex data completely understandable, and it does it without ever distorting the evidence. So the absolute most common mistake researchers make is sitting down and thinking, I really want a bar graph, before they've even articulated what they're actually looking for. Please don't do this. Start with the research question, not the graph. Your very first thought should always be, what biological question am I actually trying to answer here? Only after you nail that down do you choose the visualization. To make this easier, we can use a simple framework. Start right up at the top with your core research question. From there, identify your specific variables, and then figure out the exact relationship you're examining. It's only down at step 4 that you actually pick the appropriate visualization, which then finally unlocks your interpretation. If you try to skip straight to picking a chart type, the whole structure just collapses. As you can see here, different questions map directly to highly specific visual tools. Asking how much or how many? Well, a bar chart is engineered perfectly for that. But what if you're asking, how does something change over time? Now you're going to need a line graph. Are two variables related? That demands a scatter plot. How do different groups vary? You'll definitely want a box plot for that. The takeaway here is that different visualizations are designed from the ground up to answer entirely different biological questions. Let's look at a really specific example. Say you're investigating the distribution of leaf lengths in a particular plant species. You wouldn't use a line graph for that, right? The biology naturally dictates a histogram to show the frequency of those links, or maybe a box plot to clearly lay out the median and the spread. The key is that you are letting the biological question dictate the graphical format, not the other way around. Here's the thing about AI graphing. It is rarely a one-click magic trick. It requires a clear, step-by-step, iterative progression. You might kick things off by telling the AI, create a graph comparing plant biomass among treatments, and it'll probably give you something pretty basic. So then you push it, show the individual observations. Next, add the median and variability. After that, label the axes with measurement units. And finally, you tell it to make the figures suitable for an academic presentation. It's a conversation. What we just did there represents an absolutely vital mindset shift. You have to adopt an active workflow. Generate, evaluate, and then refine. Whatever you do, avoid that passive, dangerous trap of just prompting the AI and blindly accepting whatever it spits out first. Remember, you are the scientist here. The AI is just the one holding the paintbrush. We need a stark reality check here. AI can absolutely generate a graph that is visually stunning, but completely scientifically misleading. Current AI models are honestly incredible at graphic design. They will happily hand you a chart with gorgeous colors, sleek grid lines, and a hyper-professional layout. But don't forget, graphical design choices directly influence scientific interpretation. If the AI aggregates data incorrectly just to make a chart look a little cleaner, it has fundamentally distorted your evidence. So whenever an AI generates an aesthetic masterpiece for you, you have to step back and ask yourself, is this beautiful graph actually correct? Are the correct variables even being shown? You have to pierce right through that visual polish and aggressively interrogate the underlying science. To help you do exactly that, you should run through this essential checklist every single time. First off, verify the basics. Are the units accurate? Is the graph type even appropriate for your initial question? Then, hunt for distortions. Has the data been improperly aggregated? Are there missing observations? Finally, double-check the framing. Did the AI truncate an axis just to exaggerate a trend? Is uncertainty properly represented? And does the caption actually match the biology? You really have to verify every single one of these points. This is our ultimate roadmap, taking you all the way from a question to an insight. It kicks off with your biological question and the actual data you have on hand. Once you have chosen the right visualization for that question, that is when you finally bring in the AI to generate it. But the work doesn't stop there. You still have to actively refine the clarity, rigorously validate the graph against your original data, and finally, interpret the biological pattern. If you follow these 7 steps, AI stops being a liability and becomes a massive amplifier of your scientific rigor. Which leaves us with this final thought. You now know that visualization isn't just decoration, it's a core analytical tool. You know exactly how to match questions to graphs, how to iteratively prompt your AI, and how to rigorously validate its outputs so you don't get fooled by misleading beauty. So take a look at your own research. Look at your own spreadsheets. How will you use this 7-step AI workflow to transform your raw data into clear, validated scientific insights? The tools are right at your fingertips, and now you've got the methodology to match.
Because here's the fundamental shift we have to make. We've got to stop asking, what is the best AI? Honestly, it's just the wrong question. It leads to so much frustration and totally inefficient workflows. Instead, I want you asking, what tool is appropriate for the task I actually need to perform right now? That one simple rhetorical question, it completely flips the script. It takes your focus completely away from the tech hype and plants it firmly back where it belongs, on your specific biological data task, your specific data set, and the actual output you need to achieve for your research.
So what does this toolbox actually look like? Well, we basically have 4 main pillars for biology. You've got ChatGPT, which absolutely excels at explaining concepts and summarizing literature. Then there's Gemini, which is perfectly tailored for analyzing your data right where it lives in spreadsheets. You've got Claude, positioned for deep reasoning and handling those massive long documents. And while we aren't going to deep dive into it today, Google Colab combined with Gemini is sitting there for coding reproducible workflows. Think of these not as competitors fighting for your subscription, but as completely different, specialized instruments in your laboratory toolkit. ChatGPT is basically your go-to instrument for conversational data analysis. So, when should you actually pull this tool out of the box? Well, you use it when you need a sounding board. Say you're brainstorming ideas, or maybe you're totally overwhelmed with literature and need it summarized fast. Or you just need to explain some really complex concepts. Because of its highly conversational nature, it acts almost like this tireless research assistant sitting right across the bench from you, ready to talk through the data at any time of day. So what does this look like in practice? The sequential methodology for ChatGPT is pretty straightforward. Step 1, you just upload your biological data set, let's say a CSV of bulk RNA-seq read counts. Step 2, have the AI inspect the variables and check for data quality, flagging any weird inconsistencies or missing values. Step 3, prompt it to analyze patterns and summarize the statistical results. Step 4, have it generate charts and basic visualizations based on those exact patterns. And finally, Step 5, and guys, this is where ChatGPT truly shines, ask it to explain those findings in plain language. You can take a raw, dense biological data set and literally walk it step by step through inspection and analysis, all the way to a beautifully drafted explanation that's ready for your next presentation or paper. Gemini is the absolute go-to solution when your data already lives in a spreadsheet. Look, one of the biggest friction points in data analysis is migrating your data from one platform to another just to run a quick analysis. It completely breaks your flow. Gemini prevents that need to forcefully migrate your data elsewhere. It meets you exactly where your data currently sits. And the crucial point here is how systematically Gemini handles these tasks. First off, it lets you work directly in your spreadsheets. Second, it can automatically generate insights from the data sitting right there in those cells. Third, it completely automates the mundane stuff by suggesting the exact formulas and summaries you need. I mean, no more spending 20 minutes Googling how to write some complex nested formula for your assay results, right? And fourth, it can create charts directly in Google Sheets. It takes the immediate environment you are already comfortable in and just supercharges it, extracting instant insights without the heavy lifting.
This dichotomy here perfectly illustrates our whole toolbox concept. On one hand, Gemini is highly specialized for spreadsheet-based data insights, quick formulas, and immediate chart generation. On the other hand, we have Claude, which is designed for deep reasoning, complex analysis, and handling much longer documents. So if you need quick math and charts on a grid of numbers, you use Gemini. But if you've got an extensive data set paired with dense scientific literature that requires some serious intellectual heavy lifting, well, let's look at our final tool.
Where ChatGPT is conversational and Gemini is spreadsheet-focused, Claude is your deep reasoning engine. It absolutely excels when you need to analyze datasets right alongside longer documents. If you have, say, 50 pages of supplementary methodologies paired with a massive dataset and you need to somehow synthesize the two, Claude is the instrument you reach for. Plus, it is exceptionally strong at outputting generated code or creating really detailed visualization plans based on that complex synthesis. Let's detail those capabilities just a bit more. Claude doesn't just read the data, it supports a much more rigorous, reasoning-heavy research workflow. It can ingest a vast amount of context at once, both your data and your text. It can support deep, nuanced reasoning and explanations that go way beyond surface-level summaries. It's also a fantastic partner to generate or refine analytical code, especially if you're building out a custom pipeline. And it will help you thoughtfully plan out your final visualizations and outputs. Bottom line, it's the tool for the complex, multifaceted problems that require intense, cognitive heavy lifting.
So, we've seen the conversational agility of ChatGPT, the spreadsheet-native power of Gemini, and the deep reasoning muscle of Claude. Which AI tool will you trust with your next biological dataset?
So, we really can't talk about advanced AI frameworks without nailing down this baseline first. AI, no matter how mind-blowingly advanced it gets, absolutely requires clear, purposeful instruction from you. You're the pilot here. Prompt engineering isn't just, typing a quick question into a chat box, crossing your fingers, and hoping for the best. As defined here, it is literally an art and a technique. It needs structure. It requires you to write out super clear, purposeful instructions. Why? Well, because the goal isn't just to get some random output. The goal is to get a highly meaningful output that actually moves your specific academic work forward.
Let's look at this prompt recipe workflow. I love how it breaks an effective prompt down into 5 distinct ingredients: role, task, instructions, context, and input. Think about it. If you just toss an AI an input, you're going to get a really generic, boring response. But if you assign that AI a specific role, tell it the exact task, give it step-by-step instructions, frame it all up with the right context, and then feed it your input, boom, you've created the perfect prompt. Honestly, this specific workflow is exactly what separates casual, amateur AI usage from real academic-grade generative AI results.
This brings us to what is hands down the biggest trap researchers are facing right now, letting the software dictate the science. Let's contrast these two approaches side by side. Over on one side, you have the technology-first approach. This is when you open up some flashy new AI tool and think, wow, what can I do with this? It is such an easy trap to fall into, but it will completely mislead your analysis. You end up bending and twisting your research just to fit the limitations or the cool little tricks of that specific software. Now on the flip side, we have the methodology-first approach. This is where you start with your core research question first, and only then do you go out and find the tool that actually serves that question.
Check out this step-by-step workflow recommended by our sources. Notice exactly where the AI tool comes into play. It is not step 1. Step 1 is your research question. Step 2 is identifying your dataset. Step 3 is figuring out your analytical need. Like, what actually needs to be computed here? Only when you get to step 4 do you actually select the AI tool. And then finally, step 5, you analyze and visualize. This workflow proves a fundamental rule. Your methodological decision absolutely has to come before your technology decision.
Listen, if you take away just one single phrase from this entire explainer today, let it be this rule of thumb. Choose the tool to fit the question, not the other way around. It's just an incredibly powerful, memorable anchor for your daily work. Whenever you catch yourself getting distracted by some new software update or a trending platform, just repeat this quote to yourself to realign your focus right back to the science.
Now that we've got our methodology and our research question locked in as the starting point, let's get practical. I want you to actively think about this for a second. How do you currently choose the right AI tool for your specific research needs? Do you just grab whatever is trending on social media? Do you just use whatever your colleague down the hall is using? Relying on convenience just isn't going to cut it for rigorous research. We need a highly systematic way to filter through all the noise.
And that systematic filter is the TDSOR acronym. It stands for task, data, skill, output, and risk. These are the 5 core pillars you absolutely must assess every single time you consider bringing a new AI application into your workflow.
Let's unpack these pillars a bit. First up is T for task. You have to ask yourself, what exactly am I strictly trying to accomplish here? Next is D for data. What kind of data are you working with? Are we talking text, numbers, images? Then we have S for skill, and this one is huge. How technically comfortable are you? Please do not pick an AI tool that requires complex Python coding if you don't actually know Python, right? Pick a tool that matches your current skill level. Next is O for output. Do you need statistical tables, a specific type of graph, raw code, maybe an interactive dashboard? And finally, R for risk. Are there privacy, confidentiality, or research integrity concerns? I mean, if your dataset contains sensitive patient info, your risk assessment instantly eliminates like 90% of the commercial AI tools out there. You have to select your tools based on this exact combination of task, data, skill level, output needed, and risk.
Okay, so you've selected a tool using our framework and you've generated a result.
Awesome, but you still have to evaluate that output. And this comes with a major warning from our sources. Evaluate the tool, not just the graph. Seriously, do not compare AI tools based solely on the final pretty image they spit out. In fields like biology, chemistry, or economics, getting blinded by a sleek, modern-looking visualization is a massive mistake if the actual mechanics running underneath that graph are flawed or biased.
To protect against that exact mistake, our sources give us this incredibly comprehensive 7-point evaluation checklist. It runs through accessibility, data support, analytical transparency, reproducibility, visualization flexibility, privacy, and finally, cost and institutional access. Going through this list is how you guarantee that the tool actually supports rigorous academic standards rather than just being a fun toy that generates eye candy.
Let's zero in on a couple of the most vital questions from this checklist. Look at point number 3, analytical transparency. The guiding question here is, can you understand how the result was produced? Look, if the AI is acting as a total black box and you literally cannot explain the methodology to your peers, you just can't use it for serious academic work. Full stop. Now check out point number 4, reproducibility. Can the procedure be repeated? We all know that in science, if a result cannot be repeated, it's essentially worthless. This checklist forces you to take a hard look at whether participants can actually use the platform, if it handles your data sizes, and if that final graph can be tweaked and refined. This right here is the true difference between casual AI use and actual academic integration. You've got to move from asking which AI is best to asking which tool is fit for purpose. Because the truth is, there is no universally best AI out there. There's only the methodology that fits your specific research question, your specific data, and your specific context. So as you gear up for your next project, ask yourself, are you just chasing the technology or are you actually letting your questions lead the way?
Before we can ever truly trust the output of an AI system in a lab or clinical setting, we really need to define exactly what it gets wrong. These are the 6 primary failure modes you're inevitably going to encounter when relying on AI for data viz and analysis. We're talking hallucination, incorrect analyses, misleading graphs, privacy risks, reproducibility issues, and the heavy burden of verification. Mark my words, if you're using AI, you are absolutely going to encounter every single one of these. Let's step back into the lab for a second and look at the most deceptive hazard of the whole bunch. This is what happens when something totally plausible is actually entirely fake, and it's frankly the single biggest threat to your data's integrity. So what exactly is a hallucination in this context? Well, it's not just a simple calculation error or a random bug in the code. It's a fabricated scientific explanation designed by the algorithm to look incredibly convincingly real to a trained researcher. It's going to generate answers that sound highly plausible, but are completely false. I mean, right down to generating fake citations and inventing biological patterns that literally just do not exist. An AI will happily invent explanations, fabricate numerical values, and see patterns that aren't actually there, especially if you let your rigorous scientific skepticism slip even a little bit. Remember, the machine is ultimately just designed to predict the next logical word in a sentence. It is not designed to conduct peer-reviewed science. Let's see how this actually plays out in a real-world scenario. Imagine your digital assistant claims a sudden biomass increase in your sample is due to nitrogen fixation. Now, that sounds perfectly logical, right? It totally aligns with general biological principles. But here's the kicker. Your actual dataset never even measured that variable. The AI just connected 2 concepts that often appear together in its training data and completely ignored the actual boundaries of your specific experiment. You really have to anchor yourself on this fundamental axiom. Plausibility is not evidence. Just because a machine's hypothesis sounds biologically plausible, and just because it uses all the right terminology and beautiful formatting, does not mean it's empirical evidence. At the end of the day, it's merely a statistical guess. We've got to immediately address the silent hazard here, which is leaking highly confidential biological and clinical data. You have to actively protect data privacy, confidentiality, and ownership. You simply cannot upload sensitive biological or personal data to public AI systems without explicit authorization. Think about it. When you type a prompt into a public AI, you're often just handing that data straight over to the model's servers, potentially forever. You should never, ever feed these 4 specific types of sensitive data into an unapproved OpenAI system. We're talking identifiable participant data, sensitive health or clinical information, confidential lab results, and of course, your own unpublished proprietary datasets. Handing these over to a public model isn't just a mistake, it's a massive violation of research ethics and institutional policy. I like to use this simple, uncompromising mental check before analyzing any data. Ask yourself, if you wouldn't post this dataset on a public web forum for the whole world to see, should you upload it to an AI? The answer is a resounding no. You really have to treat that AI prompt box with the exact same caution as a public megaphone. Remember, unauthorized uploads are irreversible breaches of our scientific and ethical duties to our subjects and our institutions. You've got to think carefully before uploading anything. Protect privacy, confidentiality, and data ownership at all costs, because once that data is in the model, you cannot easily get it back. So, if the AI hallucinates and poses all these privacy risks, how do we safely harness this visualization tool without falling victim to its fundamental flaws? Well, we have to implement a strict, non-negotiable verification protocol for every single output before it ever enters our research or our presentations. Every important AI output absolutely must be checked before it is accepted. No exceptions. Consider this your new standard operating procedure. First, you allow the AI to generate the visualization or analysis. Then, you step in to strictly check it against the raw numbers. Next, you validate the methodology. And finally, you, the scientist, interpret the biological meaning yourself. More specifically, you need to execute these 4 actions every time. First, compare the AI's output directly with your original data. Second, review the statistical method it shows, like did it actually use the right test for your distribution? Third, test the biological plausibility, but remember our earlier rule. Plausibility isn't enough on its own. And fourth, apply your own trained scientific reasoning to ensure the biological claims actually hold up under real-world scrutiny. Ultimately, despite all the insane processing power and rapid graph generation we have access to now, the burden of truth rests exactly where it always has in science, with a human researcher. I want you to take this overarching thesis into your modern lab workflow. AI may generate the analysis, but the researcher owns the conclusion. AI is a phenomenal tool for generation, absolutely, but your human intellect, your years of training, and your ethical compass are the sole owners of the scientific conclusion. So as we integrate these incredibly powerful systems into our daily work, I'll leave you with this question to chew on. If an AI creates a beautiful, perfectly formatted graph that fundamentally misrepresents your data and you publish it, whose reputation is actually on the line?
I will end my presentation. I would like to give this concluding remarks using the acronym BIOTA. Let's begin with letter B. Begin with the biological question. Let the research or learning problem drive the use of artificial intelligence. I. Inspect and understand the data. Check quality, the context, the variables, and limitations before analysis. And O, optimize AI as an assistant. Use the right tool for analysis, visualization, explanation, or synthesis, which I presented to you the different tools. And then T, test and validate the output. Verify statistics, the visualizations, interpretations, and claims. And last but not the least, letter A, act responsibly. Protect privacy, document the process, and keep human scientific judgment central.